Swayam Prabha. Digital India. Educated India.
Hello students, welcome back to the organ printing course. So in this lecture we'll discuss what is required and how to print an organ. So here we'll see the different approaches required for printing an organ, or even what are the different strategies. Before that, what is an organ, what are the different structural characteristics of a particular organ, and what is required, what are the things that we need for printing an organ. So we'll discuss all these things in this lecture. Let us first start with the very basic question, that is what is an organ. And if we understand the structure and function of a particular organ, then probably we can design or develop the strategy to bioprint that particular organ in an effective way. So that's why understanding the structure and function of an organ before actually going to bioprint that is very important. And in this lecture we'll discuss that thing. So organs are, if you see, human body composed of different types of tissues and organs. So multiple organs work in simultaneously and in tandem, like together, to run our body, right? So that's why, so they have all the different organs, they have a typical 3D structures. So like if you see each of these organs, they have a characteristic, or they have a detailed structure, very complex. Sometimes some of the organs are very complex in nature. So they have a typical micro and macro structure that is required to fulfill the function of that particular tissue. So they have a characteristic micro and macro structure for function of that particular organ. Like heart is a 4-chamber organ where it receives the blood and then pump the blood to the whole body, right? So that's why heart has a typical structure of the heart is like that. Similarly, liver has a typical architecture, and if you typical macro structure, and if you see the microstructure of the liver, then they are composed of hexagonal units that is called lobules, and so millions of these lobules are connected to each other. Then the whole liver develops, because the main function of the liver is the metabolism of the food, metabolism of the food and drugs that we mostly that we ingest, and that then they produce this thing, they produce the energy, then they produce the different other components that goes to the blood, and that goes to the all the other organs and tissues of the body. So typically each of these tissue, they have the specific or characteristic micro and macro structure, and we need to understand that before actually we delve into bioprinting of all this, all that particular tissue or organs. Another thing is this: if in our body, all the tissues and organs, if we can divide them based on their cellular and acellular components, then we can actually basically find out that the all the tissues and organs, actually they have multiple cell types. And also there are other things that is outside the cell, that is the present, there is the more material aspect, that is extracellular matrices. So our body, if we can divide our body into two parts, there's the cellular components where all the different cell types will be there, and then the acellular component, that is the extracellular matrix, So the extracellular matrix is basically, it is composed of different types of proteins, carbohydrate, and inadverse tissue fat, and other type of material. And then it provides the housing of the cells. It also supports cell attachment, proliferation, migration, so the cellular function. So all these things are actually Extracellular matrix provides all these things. So that's why there's all the different tissues, but the extracellular matrix composition and the composition can vary from one tissue to another tissue, and also the proportion of all these different components can vary from tissue to tissue. Like suppose uh, that the tissue, one tissue, another tissue, suppose they have similar type of extracellular matrix components, but the proportion can vary. Also, the arrangement of this all these extracellular matrix components, the microstructure is typically that can vary. Right? Other than that, most of the organs they have the various compartments, and they also perform different functions. Something like, suppose pancreas does both exocrine and endocrine functions. So but there are separate compartments to perform these functions. Suppose the exocrine functions, they produce different types of enzymes, and that can help in digestion and absorption. But the endocrine function, where the hormones are basically produced, and those hormones basically function, that the different, like insulin, glucagon, all these hormones are produced by the this organ, and then it basically this it helps in doing, like sugar metabolism and for other thing, it can help in this all these things. So that's why, so different organs, they have a different compartments for passing. Suppose kidney had different compartments, like this glomerulus, Henle's loop, the collecting tubule, all these things, ascending tube, and this descending one, all these things. So different components are present, and they all function different things. So we need to understand the various compartments of these things, of this particular tissue, and what are the different cell types present in that particular compartment, because that is very specific for a function of that particular tissue. So that is very important for their understanding. Right. Now, other than that, also this all this structural elements of this or particular organ, there is a complexity. So there's some amount of complexity present within the all the different types of structure elements, that can range from micro to nano. Right? So different structure elements, suppose the different extracellular matrix component. So they have a typical microstructure, also they have a typical nanostructure. So this, starting from this, even if you see the structure, then there is a macro to nano, different levels of complexity present in a particular tissue, in the particular organ. So that to reproduce or to replicate that kind of detailed complexity at different length, like starting from nano to macro, that is which required for to generate or to reproduce the function of a particular organ. Right? Then other than this, that if you see different the organs, they also have some microstructural units, like liver has a liver, the microstructure and liver unit of the liver is the lobule. So lobule, which is mostly it's a hexagonal unit, where the cellular organization is very different. So that's why that all these different microstructure units, they have a unique cellular composition and organization. So that is that is very much required to reproduce when we are trying to print an organ. And all these organs, they also have a unique extracellular matrix component with nano to microstructural details, like starting from, it can be from anywhere between nano to micro, in that range. So these different extracellular matrix components, like collagen, GAG, fibronectin, laminin, there are various types of hyaluronic acid. Hyaluronic acid is present in the cartilage ECM, and most in other, like in skin and other ECM. So those different types of extracellular matrix component, they have a typical nanostructure and a microstructural detail. So that also we need to understand. And if you are using some biomaterial, then mimicking that aspect, like this structural details, is very much important for bioprinting. Also, in our body, we have a very large and very complex vascular network, by which most of the cells in our body, they are getting nutrition, oxygen, and also the waste product is getting washed away from the cells because of this vascular network. So the vascular network is the driving force of our all of our body's function, all the different tissues and organs. So this vascular network, because of the vascular network, all the different organs, they can sustain, because we have a complex vascular network. So when you are trying to 3D bioprint an organ, we need to understand, or we need to replicate that kind of vascular network in our bioprinted tissue or organ, so that that bioprinted tissue or construct can be sustained over time. They can, the cells can be viable, they can do the function, and also the whole organ can get, the whole organ can be developed. So that's why in any bioprinting task, when you start a bioprinting task, understanding the structural complexity, the structure organization, the microstructure, nanostructure, and also the organization of different cells, different cellular compositions, how they are arranged, and also what is the extracellular matrix component, that is the material aspect of the cells. What are the different extracellular matrix component they have, how those how those uh, extracellular matrix components arranged or organized within the tissue, and also in a tissue or organ, these extracellular matrix components, they are come that their organization arrangement can also vary from one part of the tissue to another part of the tissue or organ. So we need to understand those things, or we need to study in great detail about all these things before even we start bioprinting of a particular tissue or organ. So these all points are very important for for organ bioprinting, or any kind of bioprinting targeting to develop some tissues or organs. And that's why most of the time the researcher in tissue engineering or bioprinting, this they look the organ, embryonic organ development as a driving factor or strategy, how, or to understand how this embryonic organ development actually happens, and then to take clues from there to use or implement that for organ bioprinting. So if you see in your embryonic organ development, entire human body develops from a single fertilized egg. That is the very first thing we know, that when the sperm meets the ovum, and then it is, when it's fertilized, then it produces that, from there the blastocyst and other things start forming. So that's why from single fertilized egg, the whole thing, so that that divides and mature, and then again that differentiate to develop the whole type of different types of tissues and organs. So that's why organ development in an embryonic thing, it takes nano to macro approach. So that means starting from a very very small single cell to a macro tissue, macro organ, or the whole body, human body develops. That's typically a nano to macro approach. And generally the cells, the earliest cells present in that fertilized eggs, like when they are at the very initial stage, they grow and differentiate and use their inherent machinery to form a nano to microstructural organ scaffolds. That means the cells, they produce their own extracellular matrix using their own machinery, inherent machinery. The cells, they have the capability to produce a different extracellular matrix component, because by this way they can pattern, they can arrange, organize the different extracellular matrix in a nano to microstructure, so that is helpful for that particular tissue or organ development. So along with the development of this thing, along with the development of this, the cells when they are growing, they also need, they need oxygen, nutrition to sustain their growth. So that's why for providing these essential nutrients and growth factors to the growing tissue and organs when they are, in case of embryonic tissue to organ development, then capillary networks also develop along with this thing, along with angiogenesis. That means angiogenesis is a sprouting up new blood vessels from the existing one. So that actually helps to grow the tissue, that help to provide nutrition and nutrition, oxygen, growth factor to the growing tissue or organs. Another interesting thing happens in case of embryonic organ development is different organs they develop simultaneously and in tandem. That means all different tissue. If you see in the embryonic organ development, that is from the very first, very earlier tissues, very earlier few cell types, then the whole different tissues or whole different organs develop simultaneously and along with each other. So that is very much important for to understand, and also very important to how basically what are the different forces, what are the different signals that generally help in this process, like for this organ development. We need to understand that so that we can, if we can implement that in our bioprinting, then that can be a major breakthrough, and that can be a major advantage for functional tissue or organ development. And other things is like this, the meso and macrostructure anatomy organizes as the organ prepares to perform its intended function. That means, for as we have also discussed in our earlier lectures, that because all the typically different tissues and organs, they have a characteristic micro and macro structure that is required to fulfill the function of that particular organ. That's why this, whenever this the structure of the tissue or organ develops over time within the development of this in the embryonic development stages, that time this meso and microstructure anatomy, they organize it themselves so that that function of that particular tissue or organ can be performed. So this is the embryonic organ development always helps to understand how the tissue development process actually happens, and we can basically mimic, if we can mimic that kind of process, that is very helpful for our organ development. Now let us understand what are the things if we are trying to do for bioprinting, then what is the what are the things that is this is that one should consider for organ printing. Like very first thing, so the histology and anatomy in general, that is very much important to understand, like the how the what is the histology of the tissue, that means what is the structure of the tissue, how the cells are arranged in that, what is the anatomy of that particular tissue. So we need to understand that first, and then accordingly we have to figure out what type of cells to be used, like what are the different cell types present and how they are arranged. All these things we need to understand so that we can actually basically for bioprinting, as you know, for bioprinting cells are the primary primary requirement. That is because without cells we cannot print a tissue or structure. So the cells, we have to understand what are the different cell types present and how they are arranged, and we have to get those cells, or we have to use those cells for our bioprinting work. The next thing is the conservation of the structure and function of a particular tissue or organ. As we have discussed in the last slide, then each of these tissue, they have their own structure, and because of this structure, the function generally develops. That's why what type of scaffolding should be printed and what technology should be used. That is the next thing you should answer, and is next thing you should think. Like different scaffolding techniques can be. So what scaffolding technique means, with bioprinting, we can basically we can print different types of structures. That is helpful for that. So that is helpful for developing of the particular tissue or organ. So that structure we need to suppose we need to print. Then what type of what technology should be used? Because we have discussed in our earlier bioprinting class is this, that different types of tissue structures can be or tissue structures or architectures can be printed by different bioprinting technology with with starting from low to high resolution. Something like in lift bioprinting or inkjet bioprinting, the very high resolution structures can be printed, and extrusion bioprinting maybe probably the low resolution structures can be printed. So that's what we need to also understand what type of technologies required to fulfill your target tissue or organ bioprinting. Similarly, when we are thinking about developing the nano or macro approach for human organ development, then that's what when then we also have to do this, recreate that kind of structure, nano to macro structure with nano to micro architecture or structures. So different biomaterials can be employed to mimic that nano or microstructures to replicate the nano and macro approach for human organ development. That means because biomaterials they have a typical structure, and different biomaterials, if we can study them well, then we can understand the different biomaterials they have their own microstructures or nanostructure. Something like, suppose in our tissue or organ, suppose some organ, the collagen is a biomaterial. Collagen is a material protein that is present in most of our tissues in the ECM, extracellular matrix. So that collagen, typically the structure of the collagen, it is like it has a nano dimension, typically it's a nano dimension, and then they produce the collagen fibers that is in micro dimension. So we can use the several biomaterials that can mimic that nano or microstructures of that particular tissue, and then so that we can replicate the nano and macro approach for the human organ development. Other than this thing, the cells basically in the when you put the cells together, they generally they have this intelligence so that they can assemble together, they can drive the tissue formation. That means the cells can assemble, they come together, they produce their own extracellular matrix, and then they can build the tissue. But certain essential cues are required to guide or facilitate that cellular assembly so that a particular functional microstructural elements of a organ development or of a tissue development can be possible. So this is another thing where also biomaterials can help to provide that guidance students for the cellular assembly. In certain cases, the bioprinting strategy can guide the cellular assembly. When we put, suppose in case of inkjet printing or in case of laser assisted bioprinting, we can do this thing. We can provide the cells with certain, suppose if we print the cells in a certain pattern or these things where they can put them close to each other, suppose I am creating a spherical pattern, so then what will happen, these cells they can assemble with each other, they can assemble to the next one, the cells can sense the presence of another cells in the nearby, and they can assemble together, and then they can produce this functional microstructural element units, and that can help into the tissue building. So the proper placement of biomaterials is necessary to direct cellular self-assembly and organ development. So that's why when you are working with bioprinting, the placement or positioning of this biomaterial in a particular coordinate or in the tissue structure is very important, so that can guide or direct the cellular self-assembly or organ development. So these things are very important for to understand, to consider while going for organ printing. Now the other thing one should also decide or choose what type of bioprinting modalities to be used. Like you know that different bioprinting modalities they have their own advantages and disadvantages, and we have discussed in great detail in our earlier study in our bioprinting lectures, where we have seen that what are the capabilities or limitations of all these different bioprinting modalities. Suppose some bioprinting technology where the resolution is very high, but the speed is not that great, or that cannot be used for printing a centimeter scale size tissue. But certain bioprinter where the resolution can be is not very high, it's a kind of low resolution structures, but the we can definitely reach the centimeters level tissue structure. So we need to consider all these things. But most all these things, bioprinting technology, mostly they take a macro to micro approach for organ development, because where we are starting with, because we are positioning the cells, or we are positioning biometrics, we are patterning biometrics in the micro scale to recreate the geometry of the or microstructure of that particular organ, so that we can come up with a macro level tissue. So that's why bioprinting, basically in all bioprinting approaches, we take this macro to micro approach, where we want to create something macro, but we start from micro, so that we can come up with this thing. And most of the time that is done by, suppose we can, with the by different bioimaging modalities like CT, MRI, we can actually image or capture the three-dimensional geometry of that particular tissue and organ, and from there we can again recreate the anatomy of the tissue and organ with a, from the same CT or MRI data, convert them into a CAD format. That means the CT or MRI that is available in DICOM format. From their DICOM format, we can convert them into STL format by using some CAD softwares, and then we can actually we can design that CAD model, so that that model can be used for generating the or 3D printing, bioprinting the that particular tissue structures. So from the CT MRI, that is the data, that 3D image data that is available in DICOM format, we convert them into STL format in CAD, and that STL data can be used for bioprinting, directly for bioprinting of that particular tissue structure. So there, that microstructure resolution can be preserved, and we can use those things for generating, recreating, or reproducing that microstructure of the particular tissue or organ. So that's why bioprinters, they replicate the micro and mesostructure resolution by utilizing the CAD models. The CAD mod They use that CAD models where these features are preserved in the CAD model, and that CAD model, when you use that with the bioprinters, then we can replicate that kind of macro or mesostructure resolution. Certain bioprinters, they are where the resolution is not that great, but they can work on, or where by which we can work on that centimeter level size of tissue. But certain bioprinting modalities where the resolution is very high, something like 20 micron or so resolution, we can definitely reach using with inkjet or lip bioprinting with very high, so that there we can basically place or position the cells in a very detailed way, or in a particular required coordinate, where they are, like, suppose they are very much required to be printed. So that kind of, we can control the positioning of these cells at a certain location, so that we can create that localized environment of the particular tissue structure. That is very much required for bioprinting of this thing. So that's why you need to select a particular type of bioprinting for a particular type of strategy or implementation. Highest resolution bioprinting methods are also limited in replication the microstructural anatomy with high fidelity. Though different bioprinting, they can do this job, like they can be used for microstructural detailing, or also to bioprint macro tissue structures, but there are some limitation where even with the highest resolution bioprinting methods also, we cannot replicate the microstructure anatomy with high fidelity. Something, because the tissue, their microstructure details are sometimes it is too complex, that we cannot actually recreate that microstructural complexity with even the highest resolution bioprinting methods available. But that is the technological limitation, and probably there's some research work already going into that direction, whereby which we can basically develop some strategy by which we can be able to recreate that complexity, microstructural complexity of that particular tissue structure. So for bioprinting, so this is very important. For bioprinting, we take a macro to micro approach. For biomaterials and self assembly, where nano to meso approach can be used, and but if both these things can must work in harmony. So that with bioprinting, when we are working with bioprinting, using certain biomaterials or certain cell different cell types, then we should work in that those approaches should work in harmony, so that finally a macro level of tissue can be bioprinted with microstructures having nanostructural details. So that is very important for organ function. And if you see more and more of this organ to organ microstructure of the organ, then you will be you will be amazed to see the detailing that is present, and that is very much important for that organ level function of that the function of that particular tissues organs.